really fitting that we can highlight some of the work, some of the research that's being done all around us. So we are going to welcome Melissa Simon of Rhode Island Commerce, or Rhode Island Commerce Corporation, and she is going to be moderating a few short research-driven flash talks. We have John McMichael from Beechtree Labs, Angela Slitt from the University of Rhode Island, Maria Grazia Ruocco from Brown University, and Lenny Moise from EpiVax to give these presentations. There will be plenty of time for a little Q&A. Throw your comments again in the chat. We want to hear from you. So with that, I will pass it over to Melissa. Thanks for joining us.
Quick intro of myself. So for those of you who don't know me, Melissa Simon, I work with Rhode Island Commerce on the business development team focusing on life sciences. So my role with Commerce is really to help grow the life sciences ecosystem in the state, both by supporting local R&D and innovation, and also trying to go out and tell the Rhode Island story outside of the state and attract more innovative companies and entrepreneurs to relocate here. My background's all in life sciences, so I did a PhD in biomedical engineering and then also worked in university tech transfer and pharmaceutical commercialization. So great appreciation for all the work that our speakers are going to be doing. I also had the opportunity earlier this year to work on the state's COVID testing response team, help lead our strategy early on. So again, great appreciation for the complexity of this virus. I left the lab about 10 years ago, but have a great admiration for those of you who are our speakers who are still working tirelessly to try to do innovative work in the lab and help us combat this pandemic. So I'll kick it over to our panelists now to do their introductions and talk through the work that they're doing. So we're going to start with Angela.
Hi, everyone. My name is Angela Slitt, and I'm a professor at the University of Rhode Island College of Pharmacy. And today I'm going to give you a flash talk about the development of the Quanti-COVID test to develop SARS-CoV-2 mRNA. So before I begin, I just wanted to acknowledge some of the partners that have been involved in the assay development. The University of Rhode Island, and that role has been assay development and improvement, helping license know-how, and providing financial support. Thermo Fisher Scientific with regard to the assay supply chain, assisting us with the resources to design scale-up plans, as well as the ability to lend advice for commercialization. Biotechna, they're currently allowing us to license our existing technology. And then lastly, Dominion Diagnostics currently is supporting us as a potential CLIA sponsor. So I'm just gonna say something up front because I feel like this has been a long journey. So there are 4 things. There are things you know. There are things you know you don't know. There are things you know you don't know. I think I just said that one twice by accident. Anyways, the last one is the things that you don't know that you don't know. And my husband had brought this up to me. He was quoting some Army leadership training. And really, I bring that up because it's like, this is a really dynamic time. And really at the drop of a hat, really the dynamics of everything can change. And for any of you who've been on this, we feel like we're on some sort of pandemic roller coaster of ups and downs along the way. And I picked this one in particular because these individuals have masks on. And I think the roller coaster is really, is great when you're innovating, but then it becomes even more challenging under the conditions of a pandemic. So I just want us to think back to late February 2020. Rhode Island was one of the first states to have a COVID-19 case in the country. At that time, the aspects of the virus were largely unknown. There was palpable fear. I think at least us in the Northeast, we could track back to some individual close to us who either had the virus, had been exposed, was in quarantine. There was just a tremendous number of uncertainties. And we also saw that shortages were starting to occur. And this is just a picture that I took from our local Stop and Shop, kind of showing an empty toilet paper aisle and just how we were all kind of competing for the necessities. And really the question became, as scientists, like, were we in that same boat? How do we help? What do we do? And what's in our tool chest that could be used to combat COVID? I mean, I think in February 2020, those of us in the Northeast were really feeling this. And so now I flash forward to the summer of 2020, and this is a picture of the URI campus. At this point, we had been working on the assay, and I'll take you through a little bit of it, but there were some kind of real changes that precipitated the university's desire for me to kind of move this forward. How can we push this? And one of them was that HHS sequestered Cadell equipment that was that we had ordered, but instead was being sequestered to go to nursing homes. And now the semester is creeping upon us. And there's a realization that we need testing for 1 to 2 years. And at any point, those resources that we have in place may not be there. And we need these tests to be quick. We need them to be cheap. And we need them to give us some idea at the university level if someone is a weak positive or if someone is a really strong positive, because we're going to have to essentially figure out which students to go after first. And so while we're hoping that the top is more like this, we do know that in college campuses, we're hoping that it doesn't end up like this, but we have to think about those things. And so when we're thinking about this, we're thinking about what kind of diagnostic do we want when we're in the middle of a pandemic? Well, I can tell you that opinions change a lot. And we do know that we want a sensitive and specific diagnostic. We've talked a lot about ease of sample collection, and any of you who've had a nasal swab or a nasopharyngeal swab will tell you that they're relatively uncomfortable. So my bosses came to me and said, the upper administration, we know you have a test, you're working on it, but we want it to be from saliva. And it has to be really something that the students are going to embrace doing. And so we kind of feel like it's a little bit of saliva or bust. There are other aspects to assays that we need to consider, the time to result versus the robustness of maybe mass testing. And what I mean by that is, you know, do you go somewhere where you can get a test in 15 minutes and that's valuable versus the ability to kind of gather a lot of samples and run several thousand at a time. Like, for example, run one whole dorm in one day. Cost. As you're seeing it through the news, sometimes tests should be about $100. Other insurers are getting charged $800. So the idea of having something cheap that the university can rely on. And really what we wonder is, does the toilet paper argument apply in this situation? Do we have to worry about supply chain? Do we have to worry about competing with others for scientific reagents? Lastly, another thing to consider is a diagnostic tool in a hospital setting versus a tool that we're using for public health surveillance. They may not completely match up. And so there's been a lot of discussion about sensitivity versus time to result versus cost versus ease of collection. And really there's not necessarily one size that fits all. And this is just a picture from Providence PVD donuts. And basically, I think there's a lot of feeling of like, I'll take all of them. And so that's kind of what we're thinking about, you know, that different assays have different places and that not just one assay or one technology has to be it or bust. So there's different ways we can detect SARS-CoV-2. There's PCR, it's a nucleic acid test. It's the gold standard. It's exceedingly sensitive. Nobody's going to debate that. There's also CRISPR-Cas9 tools that exist. Sometimes they parlay with PCR. There's isothermal amplification, which is a lot like PCR. And that's typically, that's the strategy that is used by Abbott ID now. There's antigen tests. And then there's actually also nucleic acid hybridization, which I'm going to talk to you about for us. So we really went and said, could we use existing technology that we use in the lab to detect SARS-CoV-2? And we used what's called nucleic acid hybridization. And this is what it looks like on the right. It's really where you basically have a set of probes and they capture RNA, and then it can give a luminescent signal. And this luminescent signal, it can be in either like a 384 well plate, which is what we've been working towards, or it could actually be associated with a magnetic bead. And so this is not new technology. We use it in our laboratory all the time for research purposes, but it was used previously for HIV and HCV detection. And actually, Biotechna does have FDA approval to use this technology for SARS-CoV-2 detection from pathology samples. It's not a sensitive PCR, but some pluses is that we can go straight from a complex matrix like saliva into the assay. We can do this in a 384 well plate. It's using a different supply chain from PCR, and it's actually very easy to use. And right now our calculations put it for reagents at about $1 per well. So really we asked at the time is, could we quickly turn around this for SARS-CoV-2 because it's been used before? We had some projects early on. Locating reference materials to test it was a problem. Problems with understanding the virus, and then where can we measure it from? Can we get this from saliva? Do we have to go to a nasopharyngeal swab? And honestly, a lot of red tape. And in our case, we had some very complex IP landscape to clear. So some days I felt like I was running the hurdles and I was doing great, and other days I don't. So I just want to get to the point because I know this is short. But here's some data where we compared our assay against the FDA approved test that was being run by the Rhode Island Department of Health. And these were where we took RNA samples that have been shared with us that basically had a CT value of less than 32 and compared that to our assay. We shortened the time to result for our assay and basically saw a very nice concordance between our assay, which was reported in RLUs, versus CT values for 30 samples. We've since added to this data set. In our hands, our LOD is about 10,000 copies per mil. That seems very high. But when we think about the Yale Saliva Now test, that's actually also in the 6,000 to 12,000 range of copies per mil. So we're in the ballpark of another approved assay that's being used and has been funded. So where are we now? We are working with Dominion to go back and really refine what the LOD and LOQ would be from saliva samples and working with them to reproduce the results in a CLIA certified lab. We're currently out acquiring matched samples for saliva and nasal swabs from an Abbott ID Now platform. So this is us at the mobile health unit in Pawtucket recruiting research subjects. And we've thankfully cleared most of the IP landscape. We know that Thermo Fisher is willing to scale up to the assay up to 50,000 per day if we can make this feasible. And we've worked with them to get automation schemes in place, reference material in place. But at the end of the day, we want to know that a positive is a positive and a negative is a negative. So that's still where we're at. And what are our current hurdles? I'm an academic, not really trained to understand the regulatory landscape, so we still need help getting through that FDA EUA process or an FDA LDT process. Despite all the NIH calls, funding's actually been very hard to acquire, and the bulk of, for example, the NIH funding that went for the RADx Tech went to just a handful of companies, and it's really discouraging when some of those companies actually have slower times and capacity and limits of detection than your own assay does. So it's been frustrating. We're trying very, very hard. We have a countermeasures application that's still under consideration. And then also perhaps that scientists may not be always buying into something that is not PCR. And so, for example, we are comparing our data against the Abbott ID Now because a study came out in the summertime showing that really the Abbott ID Now is best situated for PCR values with a amplification curve that's a CT30 value or under. So that means like a moderate or highly infected individual. And I just think back to the Brady Bunch where, you know, Jan's always saying, Marsha, Marsha, Marsha. And so sometimes we are feeling like we're saying, hey, we have a very valuable tool. It performs as well as others. It's not PCR, and trying to convince people that maybe there is a place for this. So what to move forward? We need funding, obviously connections with other interested parties. This could be in the United States or international. We're still working on samples, and we're still working to kind of get that CLIA certified sponsorship locked in. So I just want to acknowledge the following individuals. We have a lot of people who've been involved: Thermo Fisher, the University of Rhode Island, Alan Rothman at URI, Biotechny, the URI leadership, and Melissa Simon, as well as the National Institute of Health Sciences that's given some of the funding for this. And with that, I'd like to say thank you for the invite. I hope everyone stays safe. These are some of the things that we did during our pandemic lockdown, and here's my information, and feel free to reach out to me.
Great, thank you so much. Well, we can save some of the questions for after we go through the panel, but one question that I just thought of right as you're going through, how has been your recruitment? You showed the picture of you guys out in Pawtucket trying to recruit people to give extra samples. How has that gone in the state? I'm just curious.
In some ways, very good. So we spent 2 days at the mobile health unit and we were able to recruit 40 samples. I think the challenge is that Rhode Island is doing so well at containing COVID that it's been hard to recruit people in that region who are positive. And not only that, but sometimes someone who's positive is reluctant to actually engage us and roll down their window and talk to us because they know they're positive. So that's been a challenge, but the use of the mobile health unit's actually been very successful. We've just changed other tactics to recruit individuals in other ways whom we know are positive. So we're currently working on that and hopefully being able to get those paired samples in the next week to 2 weeks.
Great. And then how far away do you think you are from being able to at least submit a preliminary EUA?
I think it really kind of depends on the resources and with which how fast we want to work. So realistically, we have everything in place. If we were to go full steam ahead with staff and everybody moving specifically on that mission, remember Dominion has other missions too. Yeah. So if we were on that mission completely 24/7, then it's a matter of weeks. But right now I think the proof in the pudding is getting these paired samples. We're working hard to get that, and then just working very hard to do those other assessments. But we have the value of these reagents have been used in the clinical realm before, so all those stability tests and things like that have been done, as well as the use with some of the automation platforms.
Great. All right, well, thank you so much. Move on to John McMichael from Beechtree, if you're ready.
We're all set. Thank you very much. Thanks for the opportunity. Beechtree is involved in developing new medicines. Our interest is in the discovery and early stage development, and we license out to bigger companies. We go through phase 2 studies, usually from time to time, phase 3. And what we're doing with the COVID-19 situation is relying on some experience we've had in the past. We are in phase 2 studies now with COVID-19. This slide just gives an example of some other phase 2s we've done. I think we've conducted a total of 18 or 19 successful phase 2 for different formulations, different clinical indications. And the bottom one on this slide, the herpes simplex, is the one that we used as a jumping off point for what we're doing now with COVID-19. That is to say, we a couple of years ago conducted 2 successful phase 2 trials using our product called BTL-TML for the treatment of recurrent oral herpes infections. We know from our past experience with that how this product works. Just shorten it to TML. We know it downregulates pro-inflammatory cytokines, upregulates anti-inflammatory cytokines, inhibits viral replication. And the 2 trials with the recurrent oral herpes showed clearly that the product is safe. And so we wondered, might it work for the COVID virus also, using the very same protocol we used in the FDA trials for herpes. Our experience anecdotally over the years has shown that TML is effective against a fairly broad spectrum of viruses. You can see that on the next slide, that we have clinical indications for these different virus entities. The TML is very potent. Everything from influenza, where we oftentimes see reduction in symptomatology within 15 to 30 minutes, and then down through other of the herpes viruses like cytomegalovirus, shingles, and Bell's palsy, and so forth, and even Ebola. It is not an antiviral that covers all viruses. Interestingly enough, it's very potent in treating the true influenzas. It's worthless in treating the parainfluenzas, the 24-hour flu. And that's for reasons we don't know. But again, because of the experience we had with herpes viruses, we decided to ask the FDA if we could jump right into a phase 2 trial with the coronavirus. In the meantime, physicians in our group treated some patients, now well over 50 patients anecdotally. These patients are not part of any formal study. They are instead just being treated as a consequence of practice of medicine. And you can see here that this is what we generally found. We could bring the temperature down pretty rapidly, certainly within a matter of 10 to 12 hours, decrease coughing, get rid of the very severe gastric pain that some patients talked about, where they felt that it was as if somebody was grabbing their stomach and just squeezing. The flu-type aches and pains were diminished. We get a return of sense of smell and taste and improved energy. What's important is all of these observations were within usually 10 to 15, 18 hours. Certainly within 48 hours, all symptoms are cleared. And some patients, in fact, one of the patients who is himself a physician, cleared in 4 hours. So we thought that with this background anecdotal information, it might be worthwhile filing with the FDA for a formal phase 2 trial. And that's what we've done. We now have authorization from the FDA to do the trial, and the trial is in fact in progress. You can see here that our entry criteria include being over age 50. We're looking for the little bit older patients, the ones that are more likely to have comorbidities, although we don't require that. We want them to be symptomatic, non-hospitalized. One of the things we want to do is keep them out of the hospital. So the way it's set up now is we try and get these patients within 36 hours of a positive diagnosis. And this is a virtual clinical trial where we never see the patients. They don't have to come to a clinic. They don't have to give blood. Physician doesn't have to go see them. It's all done by telemedicine, so they can be sequestered in their homes or retirement community, wherever it might be. We collect data from them twice a day, everything from temperature, oxygen saturation of blood, pulse rate, cough rate, and then we use what's called the SF-36, which is a quality of life survey. We do that once a day in the evening to compare how they are today versus 24 hours ago. And you can see both the primary and secondary endpoints. Of interest, and as a bit of a head scratcher to the FDA, we're looking at the primary endpoint being 48 hours after initiation of therapy, which is pretty fast compared to other products that are being evaluated. But our anecdotal experience has indicated that if we don't have them under control in 48 hours, we will not get them. But of the 50 plus patients we've treated, they've all responded positively within that period of time, and they've done so without adverse effects. So what we're doing now is enrolling patients. Our primary test site is in Salt Lake City. We expect next week to open another site in New Orleans. We are looking at a couple others, one in Tampa, one in Memphis. And again, what we want to see most is, are the patients better? Second, we want to know how long do they shed virus after initiation of therapy. This is a 40-patient study, equally divided between placebo and treatment group. Our arrangements with the FDA are making the assumption that this pilot study is finished in a successful manner, and we'll jump right into a phase 3 trial, about a 250-patient study, minimum of 4 sites. We've already identified those. In the meantime, we expect to have a emergency use authorization, and then we will license out or sell the product. So that's where we are in this project. Any information can be had from these websites, and we're glad to answer any questions that you might have. And thanks, Melissa and the team, once again, for an opportunity to present.
Great, thank you. One question I'll ask now, again, everyone else listening, feel free to throw questions in the chat. Perhaps this is known to others, but how is the idea of doing a virtual clinical trial a fairly new concept, or has that been sort of an established process?
No, it is fairly new. The FDA has specific regulations for it, so it's old enough to let the FDA get their arms around it. They're actually encouraging these kinds of trials, especially for infectious diseases, since the patient doesn't have to go anywhere and no one's exposed and the patient is not exposed to the outside environment. It's taken a while for us to work with computer savvy companies that can be able to collect and analyze these data on a real-time basis. But I think long-term, especially for a disease like this, it's really handy. And the computer company can do it anywhere in the world. We are, in fact, talking with companies internationally that are interested in this project. So we're looking forward to expanding the whole virtual clinical trial sphere.
Great. All right, thank you. All right, so we can move on to Maria Grazia.
Thank you very much for the opportunity to talk that you have given me to talk about our work. So Lab on a Chip was created as a spin-off of the University of Rhode Island by two faculty members, Konstantin Ananiastopoulos and Mohamed Faghri. They are both professors at the Department of Engineering and experts in microfluidics. While Konstantin is serving as a president, Mohamed is the chief scientific officer, and I joined the company as the business development manager. Lab on a Chip commitment towards community is to develop specific assay for point of care in vitro diagnostic for urgent medical care, underserved population, and routine testing. At the beginning of the COVID outbreak in early this year, our government were forced to lock down countries and shut down the economies. It became very clear the importance of testing, and soon if we wanted to maintain our economies running, still ensure mitigating the health risk for the population. And testing, tracing, and isolation became a sort of mantra, but most of all, it became the first goal that government and the healthcare industry wanted to achieve. A COVID testing not only has to be accurate and reliable, but also fast, frequent, cheap, and easy. According to the FDA, there are two current methods for testing COVID-19. Diagnostic tests that identify the virus. So they are known as RNA tests done by PCR and antigen tests that identify the viral protein. Then antibody tests that are directed towards the identification of the antibodies made against the virus by the patient immune system in order to fight and clear the infection. While diagnostic tests detect acute infection, antibody tests are better suited to identify past infections. Lab on a Chip technology is suited to both perform a protein test as well as an antibody test. Our solution is a rapid point of care diagnostic platform that is paper-based and therefore is cheap and frequent, can be used frequently, is based on a well-established ELISA methodology, therefore it delivers accurate results and uses proprietary valves that make the platform functioning autonomously. Therefore, it doesn't require technical expertise and any lay person can use it. We recently have further implemented our platform by the addition of mechanically operating valves. These two pictures here on the left show two types of prototype of our platform. Our platform can analyze nasopharyngeal swab of saliva and deliver an antigen test or blood to deliver serological test. The principle through which it works is basically the same. The difference lays in the reagents that are on the platform. This schematic here shows an overview of our test platform. In critical areas in our platform are are 1, 2, and 5. 1 is the site, the sample pad where the sample is loaded. 4 and 5 are the places where the reagents are stored. As the reagents are released and the sample is added, the sample goes, the sample reaches area 2, which is the conjugate pad. Here, if the virus protein is present, it reacts with secondary antibodies that are conjugated to reporters. As the liquid further travels, carries on the complex secondary antibody and antigen to 5, which is the test site. Here, the complex secondary antibody, that antigen is captured by the capture antibody. A reaction at this point will occur in the presence of this substrate released from 5, and the precipitate will form. The precipitate will be visible by naked eye as a colored line. The presence, the appearance of this colored line indicates that the samples contains the virus. Our product development is a 4-stage process: the feasibility, the development, the design development, and the clinical validation. The feasibility phase aims at demonstrating that we can identify the virus by using the paper chip and commercially available reagent. At the development phase, critical elements of the lateral flow technology are optimized in terms of time, delay, and volume. The design, development, and verification is the phase when the test becomes completely autonomous. The final version of the test is fabricated. At the end of this phase, a minimum viable product will be delivered. A few hundred of units of this product will then be fabricated and used in the next phase, which is the clinical validation. This is an extremely critical phase. Real samples are used at this point, and in accordance to FDA guidelines, clinical studies are performed. The result of these studies are then used to request an emergency use authorization. Upon obtaining this authorization, the test then can be distributed. In order to develop our test, we have established a strong partnership with local partners. We NEMICAST provided a great help in structuring this partnership. Lab on a Chip collaborates on the feasibility aspect with Professor Alan Rothman at the University of Rhode Island. Alan is an expert on infectious disease and collaborates with Axamedica. We all have heard about their capabilities on this optimization of the platform and the fabrication of the commercial product. Axamedica is also responsible for the regulatory path, and the clinical trials will be run together with the Rhode Island hospitals. I'd like to stop here and just to remind you that testing remains key worldwide, whether we have or not a vaccine. Point of care in vitro tests are rapid and safe. In many cases, actually, they are safer than other methodologies, especially for healthcare professionals, as they do not require handling and transferring of the samples. They are versatile. We plan to leverage our testing platform by combining it with a mobile app to provide quantitative measure of the test results. Initially, we plan to deliver the test to medical professionals at doctor offices, schools, and emergency room. Later on, the test will be made available at local pharmacy. People can purchase the test and perform it in their own homes. We are all aware that COVID-19 is just the outbreak of pandemic, and we know that there are other viruses that lie in the weight. Our technology can be easily adapted to face other health challenges, and next time we hope to be more prepared and adequate to respond to it. I thank you very much for your attention. I'm happy to answer your question.
Right.
I like to add something. I'm Mohammad Fagri from University of Rhode Island. Essentially, I just wanted to say this is a pretty easy platform. It's paper-based platform like pregnancy tests, but it could do ELISA on paper for the first time. So we could store a couple of reagents on paper with our technology and do sequential loading of reagents onto the detection spot via our fluidic circuit, microfluidic circuit. And as Maria Grazia mentioned, we are in the process of actually getting more funded research in this area. If people are interested to help us out, we have a promising device that could detect COVID-19 very easily. We just need funding to continue our research further.
All right, thank you so much for walking through your work. I think in the interest of time, we'll move on to Lenny.
All right, my name is Lenny Moise. I'm Director of Vaccine Research at EpiVax. I want to thank the organizers for the opportunity to speak to you today about our SARS-CoV-2 vaccine development effort. Our vaccine called EpiV-COVID-19 is directed for healthcare workers and first responders. Before we get to that, a very brief intro about EpiVax. We're a 22-year-old Providence, Rhode Island-based biotech company. We design and develop biologics and vaccines using our cutting-edge immunoinformatics platforms. Over the years, we've earned the trust of the scientific community, as evidenced by several peer-reviewed publications in the scientific literature, as well as funding from government agencies and corporate clients. Our core expertise is in predicting triggers of immunity that are found at the host pathogen interface. And that is found where a snippet of a pathogen, a peptide epitope, is presented by an MHC molecule at the surface of an antigen-presenting cell to the T cell receptor at the surface of a T cell. Knowing these triggers is important because they unleash immune processes that lead to protection against disease. In SARS-CoV-2, that can occur via T cells, which kill infected cells, or through antibody that block virus entry into cells. Most SARS-CoV-2 vaccines are delivered the antibody target, spike glycoprotein. Our vaccine is different. Our vaccine delivers these peptide epitopes that not only can support antibody development with reduced risk for antibody-dependent enhanced disease, but also stimulate the T cell component of an immune response that can kill infected cells. Now, we have computational tools that predict these peptide T cell epitopes, but not only that, we are able to discriminate between epitopes that can activate effector T cells that turn on immune responses and epitopes that could induce regulatory T cells that shut down immune responses. This is important because pathogens are known to camouflage themselves with human-like epitopes that are recognized by regulatory T cells that we developed for self-tolerance. So knowing this information, or being able to discriminate between these two types of epitopes, we select epitopes that activate protective responses and avoid those that would activate regulatory T cells. This is very important in how we design our vaccine. So with that epitope information, what do we do with it to actually make a vaccine? Shown in this slide is the strategy we take to go from genome sequences all the way to a vaccine. We start with epitope identification using our IVAX immunoinformatics platform, and then validate those predictions in vitro assays. And we go on to build vaccines using tools in the IVAX platform, and test those vaccines in vivo in an animal model to evaluate immunogenicity and efficacy. We have done this for several targets: HIV, TB, smallpox, tularemia, pandemic influenza. We're working on a universal flu vaccine as well. When SARS-CoV-2 emerged, we plugged into this process. And we started by collecting SARS-CoV-2 sequences and ran them through IVAX. In the process, we selected epitopes that are conserved among SARS-CoV-2 strains and broadly cover the MHC diversity of the human population. We then went on to actually produce the vaccine, leveraging our experience at our subsidiary, EpiVax Oncology, that produces personalized cancer vaccines. So the vaccine is composed of epitopes encoded in peptides. Peptides are a great technology for vaccines because the production process, synthesis, and the purification methods are very, very well established. Peptides have an extensive history of safe use. In addition, peptide manufacturing is highly scalable from the research or milligram scale all the way to kilograms. And there's infrastructure that already exists around the world to transfer this technology to produce millions to billions of doses of vaccine. We pair peptides with poly-ICLC adjuvant to boost immune responses. This adjuvant has an excellent safety profile in clinical trials. So where are we now? We have GMP poly-ICLC. We have made GMP peptides. We've submitted a pre-IND to the FDA and got a great response. There is no need point where we're writing the IND. What does the future outlook look like? We are writing the IND and expect to submit it in about 2 months. And we're writing it with Parexel, which helped us with the pre-IND as well as the CTAP. On the manufacturing track, our drug substance partner, Polypeptide, makes the GMP peptides. We're partnering with Mount Sinai for drug product formulation and fill and finish. And then we will start the phase 1 trial that will be performed with Mount Sinai in New York in partnership with our adjuvant supplier, Oncovir. In parallel with the trial, we will be scaling up for the phase 3 trial where we'll need more material. And they'll be working with Polypeptide and a fill finish facility that can handle larger quantities. We expect that the phase 3 trial will start on the heels of the phase 2, and the phase 2 should be activated once we get positive signal from phase 1, primarily safety signal, and we will be measuring immunogenicity as well. Now, before I finish, I don't want to leave you without any data that our EPV COVID-19 peptides can stimulate protective immunity. Like most vaccines, EPV COVID-19 aims to recapitulate immune responses that protect a natural infection. So therefore, we expect that EPV COVID-19 peptides will be recognized by people who have recovered from SARS-CoV-2 infection. And to determine that, we're stimulating immune recall in cultures of peripheral blood cells from convalescent donors using the EPV COVID-19 peptides and measuring cytokine production using an ELISpot assay or fluorospot assay. And we measure interferon gamma as our signal. Interferon gamma is the signature cytokine of the protected immune response. So what we find in ex vivo assays is that convalescent donors recognize the EPV COVID-19 peptides. In contrast, no naive patients, people who have no SARS-CoV-2 experience, do not recognize the peptides. Now, we've done a cultured assay as well after a period of time where we're expand epitope-specific T cells that are present at low frequency and not detected at the ex vivo stage. We're able to detect them after a period of culture, and as not surprisingly, we see very strong responses in the convalescent donors. We were surprised when we saw very strong responses in people without SARS-CoV-2 experience. So what this suggests is that our EPV COVID-19 peptides could stimulate pre-existing immunity to SARS-CoV-2 that is probably generated through infection with common cold coronaviruses. This is important because EPV COVID-19 can stimulate cross-reactive memory that could boost immunity in vaccination. And what I mean by that is not just the magnitude of the response, but also we may see an accelerated immune response to provide protection sooner. So I'd like to acknowledge with that our EpiVax coronavirus vaccine task force. I think the most important ingredient in vaccine development is the talent. Our founders in the C-suite, Annie DeGroot and Bill Martin, our team with expertise in project management, immunology, informatics, and finance, and our team of consultants that bring manufacturing and clinical expertise to us to help get us to phase 1 and beyond. And thank you very much for your attention. I'll be happy to take your questions.
Thanks so much, Lenny. Very, very exciting to hear a potential vaccine coming out of work in Rhode Island, since I think everyone is just counting down the days until that's available. I'll open it up if there are any questions from the audience, please chime in. I do want to address, I know Douglas, you've sent over a couple of comments throughout the course of the presentation, I think mostly focused on some questions related to availability of rapid tests. I don't know if you want to unmute yourself and maybe elaborate on some of your questions for the panelists.
Sure. So just curious about some articles I'd read about like Roger Williams University specifically. I know there was some comparison of what was happening at URI. So Roger Williams is working with a specific company and trying to do very frequent testing. Just curious if anybody was familiar with that company and that was doing the trial there at Roger Williams over in Bristol.
I personally do not, but I can look into it. I was trying to read while these were going on, and I can't multitask, but I can, unless someone else on the Zoom happens to have more information and can answer.
No.
Melissa, isn't
Yes.
Roger Williams using the Broad for their testing?
For their undergraduates, yes.
Yeah.
Is that what you're referring to, Douglas? The fact that they're...
There was a specific company that I just had seen in an article recently. It was the New Yorker. It was Homodeus, and I don't know actually what they ended up using. They may be doing multiple things. I do know just from hearsay from a student who's a friend of one of my kids that it's every other day. I think they're being tested. It's super frequent, which in the absence of an actual vaccine is how we're going to kind of be able to isolate people who are actually carrying it and do real contact tracing. If it's not done frequently, especially where people don't have symptoms, it's really our only hope in the short term.
Yeah, yeah. Okay, so I think what you're referring to is Roger Williams and I think a couple of other universities in Rhode Island as well as Massachusetts have partnered with the Broad Institute, which is affiliated with MIT, to do testing. And that sort of Broad's methodology is every other day, a couple times a week. I think the university can decide what they want to pay for to do testing. And I mean, so far I think it's going well. It's only been a couple weeks, obviously, that these college students have been back, but that's one method. There's a lot of philosophies out there around trying to do more frequent, less sensitive versus less frequent, more sensitive, and very differing opinions in that. But
great. Yeah, I've seen some comparisons. It's interesting with the private schools versus the public. There was an article about Duke University versus UNC and NC State, and that Duke has very few positive cases. They've done a tremendous amount of testing, but in terms of people being able to opt in and students' rights and also the funding available in private schools, so the rich are safer. It's a challenge there.
Yeah.
Actually, Brown University tests faculties once a week, and students twice a week. Students that and the result comes back in less than 12 hours. And they have partnered with I think MIT or Harvard, one company in Boston. Anyway.
Yeah, Brown's definitely limited the number of students back as well. So.
Question for Lenny. I'll ask unless somebody else has questions. What, sort of the $1 million question, what do you think is the future of sort of the vaccine landscape? Obviously, there's a ton of companies at various stages, at various capacities of manufacturing. Do you have a thought on what you think things might look like a year from now?
Well, that is fraught with a lot of risk. I mean,
you don't have to answer the question if you don't want to.
We predict epitopes, but with more confidence than I can predict what will happen in a year from now. But I am optimistic that we will see some vaccines approved, certainly by a year from now. But and we will need multiple vaccines, even if they're very effective. We will just need the capacity to distribute vaccines so widely that that one is not going to be enough. So we should all be rooting for many successes, and it's quite remarkable how far we've come in so short a time, and how much has been done already and how much there's left to do. I think that, if we see approvals by end of the year, early next year, then the vaccines will start to be distributed. Not everyone's going to get vaccinated on day one. And in a year from now, hopefully, we will see that a very large percentage of the population will be immunized, or at least a very large percent of the people who want to be immunized will be immunized. And we will be, getting much closer to life as we knew it before SARS-CoV-2 emerged. And it's all about timing, of course, and how efficient the distribution and the equitable access to the vaccine is made that is going to make the difference. So I'm hopeful that in a year from now, we may not be 100% back to normal, but we're going to be much closer than we are now.
And Lenny, another question for you that came through the chat. Can you talk a little bit about your plans for pre-production of vaccines prior to approval? Whether it's yours or just in general, if you have any comments on that.
Well, everything is moving at the speed of light. Companies are working at risk, and that's how we're able to, how we see that so many companies are reaching phase 3 and will be able to release vaccines so soon after they receive authorization following phase 3 efficacy trials. So I think that the capacity and the the processes are in very advanced development. And I mean, I don't know them. I don't know the stories for the different leaders like Moderna, Pfizer, BioNTech, and AstraZeneca, J&J. But I believe that they're all moving, doing everything in parallel at risk in order to have large numbers of doses ready as soon as possible to get them into the to the prioritized groups, and then build and continue building to immunize the masses of people who are behind them.
And I know we have 1 minute remaining. I guess my last question I'll just throw out to, I think Angela had to drop off, but to John, Lenny, and Maria Grazia, especially since the next panel is all of Rhode Island Bio's bioscience leaders. Is there anything that the community can do to help your efforts going forward that you'd like to quickly add in 10 seconds?
Yeah, for us, definitely additional funding will be helpful to move our project forward and to completion. Yeah, the local community is great support, and we got lots of positive feedback and help. We need extra funding to finalize the project. Yeah.
Yeah, we would like to see advocacy for funding, but also networking that would help us carry our vaccine forward even faster.
For us, for Beechtree, it's a matter of having sites for clinical evaluation of our therapeutic agent. And if Rhode Island could provide one of those, that would be great.